We're with Tad Sonstegard today, the Chief Scientific Officer of Excelligen, which is the food animal division of Recombinetics. Based in Minnesota, the firm has pioneered gene editing using the CRISPR technology, including work in pigs and cattle, identifying the POLD trait and the gene combination for heat resistance, among others. Tad, could you give us a little background on your work and your firm?
Sure. So I've been with the company since 2015, and our company's actually been around since 2008. Excelligen started in 2013, and the goal was really to use gene editing as a precision breeding tool for genetic improvement of livestock and other food animals. And the main focus of our mission to start with has been on improving allele frequencies for those traits that are involved with animal welfare. So one of those traits, as you well know, is POLD. So in this case, by introducing the POLD trait into animals, they no longer need to be dehorned, because horns can be a safety hazard both for the handler and also other livestock nearby. So what we've done is in 2014 is we had edited some cell lines and introduced a small little bit of DNA that was copied into the genome of a dairy animal that was horned. And then the nucleus from those cells was cloned by somatic cell nuclear transfer at a place called Transova. Two calves were born from that. Their founding animals were horned, but when these animals were born, they were polled. We published that work in a scientific journal, and then those animals were moved to the University of California, Davis, where they were further studied to make sure that there was no risk involved with use of this technology. And of course, what it showed is that this is nothing more than just another breeding tool that producers and genetic companies can use to introduce traits like POLD and improve animal welfare, animal well-being, by removing some of these needs for practical management practices of the past. Relative to the heat tolerance, that was a trait that I had been studying. That if you had heard of Senapole cattle, you realize those cattle were developed in the Caribbean and they were naturally tolerant to heat. So we did some genomics and resequencing of those animals when I was at USDA, and what we found is that there's a 1 base difference in a gene called prolactin receptor that shortens the protein, and so the animals that are heat tolerant don't respond the same to the sun or to a prolactin surge as normal animals do. So we introduced this 1 base deletion into another cell line from a Red Angus animal that was born in Minnesota. We took those cells down to Brazil. Those were cloned, and the resulting animal was a thermotolerant Red Angus that had a 1 base difference from a normal Red Angus, and we call her Genzel. And so she was recently featured in a Wall Street Journal video article.
We saw that video, and we'll be offering that link to our audience. Very, very interesting. Nicely done, of course, by the Wall Street Journal. And just confirm for us that those animals, the particular animals that you've talked about with the POLD gene and the Red Angus, they would breed that trait on in their offspring.
Oh, absolutely. This the use of talons or CRISPRs introduces changes in the genome that are completely stable. We're not introducing transgenes. Sometimes in the past, using old technology, transgenes were introduced by random integration into the genome, and you could get multiple copies integrated in, and those could be unstable. We're not talking about 1980s technology anymore. This is dead-on precision breeding, and what we introduce as a change into the genome is just as stable as anywhere else in the genome. So it's a pretty natural process. I will say one thing about Genzel. What's amazing is we actually can see her phenotype of thermal tolerance is when we go to visit her, she prefers to lay in the sun instead of seeking the shade, even though it's hot out.
Interesting. Earlier this month, your firm has announced some new work, a partnership to work on the TB trait to eliminate the TB in cattle. Tell us a little bit about that project and how you see that unfolding.
Right, so this is funding from a USDA NIFA grant on our side, and it's a special program they have also with funding agencies in Northern Ireland and also in the Republic of Ireland. And there's a lot of expertise both in studies on TB disease in Ireland because TB is considered still a major problem in cattle in the UK and Ireland. And then we also have a partner in Northern Ireland that specializes in stem cells and somatic, stem cells and differentiation of cells. So the idea here is that we're using this stem cell system to introduce variants that may have an effect on how well the cow's immune system can fight a TB infection. So it could possibly, some of the variants could possibly make it completely resistant, or could make them much more resilient where they can have an adaptive immunity and not shed a lot of bacteria to other animals in the same paddock or feedlot. So our goal is not to make animals in this study, but it is to test different variants that exist in nature. We plan to test about 20 of those in this cell system, and then once we find the ones that are best against the pathogen, those would be candidates for introducing into cattle to make them TB resilient or resistant.
The announcement that your firm issued talked about a $3 billion cost annually of TB in cattle, and so there's quite an impact worldwide. We just saw an announcement the state of Michigan found TB in another beef herd, again from interaction with wildlife, probably in Michigan. So this would have the potential to be of great benefit, not only in Europe but around the world.
Yeah, absolutely. Our aim in this is not only to better understand what variants help cattle become more resistant or resilient to TB, but we'll have a better understanding of the disease itself, and some of this knowledge could be applied to control possibly in wildlife species, but I don't think that we'll be aiming to do any editing in wildlife species relative to TB.
But if the cattle herd is resistant, then that would provide protection against TB in the wild.
That's the hope. That would be the ultimate goal, yeah. Sure.
Well, we appreciate this background. Can you give us any insights into some of the other possible work that you and your firm are doing?
Yeah, next month we start producing what we call castration-free pigs. And so this is another animal welfare trait to remove surgical castration or chemical castration from the production chain. And so this is, again, another research project that's this time funded by USDA FFAR, which is another agency within USDA. And it's a pretty novel funding opportunity looking directly at an animal welfare trait, and we're taking what we believe is a pretty unique approach. Of course, when you make animals that never enter into puberty, you somehow have to rescue them in order to breed them. So there's a twofold part to this study. Not only what does the edit do, but how can we use this edit to create production pigs that no longer need castration.
Well, thank you very much, Tad. We've been speaking with Tad Sonstegard, who's the Chief Scientific Officer of Excelligen, a division of Recombinetics based in Minnesota. Thank you, Tad. We appreciate you being with us.
Joel, I appreciate your interest, and I hope anyone is interested in the technology, go ahead and visit our website, and we'll provide more information if necessary.
Well, thank you very much. This has been Joel Hastings for Dairy Business News and This Week in Dairy.